Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera kepada semua yang mengikuti siaran webinar pada pagi ini. Bagi memuliakan majlis, marilah kita bersama-sama memanjatkan kesyukuran kepada Ilahi dengan membaca surah Al-Fatihah. Bismillahirrahmanirrahim. Terima kasih diucapkan kepada semua kerana sudi bersama-sama kami di dalam siri webinar anjuran bersama Jabatan Pengurusan Tanah dan Unit Jaringan Industri dan Masyarakat kali ke-6. Bersama-sama kita pada pagi ini adalah Dr. Ali Tan Ki Zuan dari Jabatan Pengurusan Tanah. Sedikit pengenalan mengenai Dr. Ali. Beliau dilahirkan di Pasir Mas, Kelantan dan memperoleh didikan awalnya di Kuantan, Pahang. Beliau dikurniakan ijazah kelas pertama Bachelor Sains Pertanian pada tahun 2009 di UPM dan memperoleh ijazah Doktor Falsafah di dalam bidang microbiologi tanah pada tahun 2010. Kini, beliau berkhidmat sebagai pensyarah kanan di Jabatan Pengurusan Tanah Fakulti Pertanian UPM. Sepanjang perkhidmatan beliau di UPM, terdapat pelbagai jurnal serta prosiding hasil kajian beliau yang telah diterbitkan sama ada di dalam mahupun di luar negara. Beliau juga turut aktif di dalam menyertai pelbagai seminar dan konferens serta turut diberi kepercayaan memegang jawatan sebagai bendahari bagi Persatuan Sains Tanah Malaysia dan Persatuan PGPR Malaysia. Hasil dari kajian beliau dalam bidang ini telah membuahkan hasil sehingga berjaya mendapatkan satu paten berkaitan teknologi mikrob dan penghasilan produk bernama Smart Enzyme UPM. Kajian beliau bukan sahaja mendapat tajaan dalam bentuk geran penyelidikan dalam dan luar negara, malah turut menarik minat pemain industri untuk bekerjasama di dalam penghasilan produk baja berasaskan bio. Kini Dr. Ali Tan Ki Zuan giat menjalankan kajian di dalam bidang penggunaan mikrob berfaedah yang menjurus kepada mekanisme berkaitan interaksi di antara tumbuhan, tanah dan mikrob bagi mempromosikan konsep pertanian lestari. Dengan segala hormatnya, dipersilakan Dr. Ali Tan Ki Zuan untuk menyampaikan perkongsiannya yang bertajuk Utilization of Beneficial Microbes for Sustainable Agriculture, Keys to Success. Dipersilakan.
Okay, thanks to Puan Atira for that brief introduction. Assalamualaikum and a very good morning to everyone. Thanks for making time for today's sharing session on the topics of soil microbes. I've been informed that the audience today came from different background and knowledge on soil microbiology. So I tried my best to adjust the content to suit and cater to everyone. And the title today is Utilization of Beneficial Microbes for Sustainable Agriculture. Basically, we are going to look at how we can make use of microorganisms in tropical soil to reduce the chemical fertilizer usage, to save our nature whilst improving the crop growth and yield at the same time. And to maximize the benefits of soil microbes to our crop, there are a lot of factors to be considered, and this sharing session will cover some, if not all, of them. Hence the title, Keys to Success. And before we begin, I would like to acknowledge and thank my mentor, Dr. Zulkifli Haji Shamsuddin and Dr. Razia Othman, and because some of the slides and informations were extracted from my fellow lab mates of the Soil Microbiology Laboratory at the Department of Land Management, Faculty of Agriculture, UPM. Now, to begin with, people always are skeptical when we talk about soil microbes, biofertilizers, and so on. Why is that so? Firstly, it's because we cannot see these soil microbes with our naked eyes. Secondly, their effect on crops is usually relatively slower compared to the chemical fertilizers. And thus, it is quite a challenge to convince the farmers to change their conventional practices and start using or utilizing soil microbes. And these are some of the questions related to this topic. why we choose to work with beneficial microbes, what is so special about these tiny little microscopic creatures, what are their benefits to our soil and plant, and what is the advantage of soil microbes over the conventional chemical fertilizer approach. And why do we choose to work with soil microbes? The first and most crucial factors is related to our soil condition. Okay, looking at this global soil pH map, we know that Malaysian soil is mostly acidic. You can see the reddish color, and generally the optimum pH for the best optimum pH for crop cultivation is 5.5. But in our case, in our soil, it is usually lower than that. Sometimes in the case of acid sulfate soil, it can goes down up to pH of 3. So based on the soil pH map, all soil are acidic. So what will be the disadvantage of acidic soil? The acidic tropical soil has low pH and it is less fertile. Why is that so? Because it contains a relatively low organic matter. Our, not, our nitrogen, a mobile element, is being leached out. When you apply chemical nitrogen fertilizers, not 100% is being taken up by the plant, and we have a relatively high amount of phosphorus in our soil, but the problem is they are being fixed by aluminum and ferrum, which are abundant in acidic soil, into the form of complexes, aluminum phosphate and ferrum phosphate. And these forms are not readily available for plants uptake. Similarly, in the case of potassium, most of our soil potassium is in the form of minerals, like for example, mica and feldspar, and thus are not available for plant as well. It can be broken down naturally through weathering processes, but this kind of processes is very slow and time consuming. So this is where our beneficial microbes came into picture. They can first fix nitrogen from the air, from the atmosphere, and supply it to the plant. Secondly, they can solubilize those complexes of phosphorus and potassium, making them available for plant uptake. So how do they do that? That will be described in the later slide. And another reason why we choose to work with soil microbes is related to the current and pertaining issue worldwide, which is the food security. As you can see from this graph produced by FAO, sometime around this year, we are going to look at the situation in which our human population is higher than what the food being produced or what the food can be produced by our in agricultural sector. So um, that is why we are facing a shortage of food, and this is a major concern, especially for net importer countries, because when producing countries are facing food security problems, they will stop exporting their produce, and we have started to see this kind of trend happening, such as in the case of India, where they decided to ban the export of wheat. So what will be the short-term solution? To increase the food production to overcome the problem of food shortage for farmers, the easiest solution will be to apply more and more chemical fertilizers, and with the hopes that they can get higher yield out of their crop. But we are not denying, yes, we are not denying the fact that chemical fertilizer works very fast. But excessive use of chemical will cause several problems, which we'll be looking into later. And we observe an increased trend of world fertilizer usage, an increase as high as 20% in the case of pea fertilizers. And this trend is expected to remain over the years. And despite despite the rapid effect of chemical fertilizer on crop, excessive use could be harmful not only to the environment but also to human health. We started to see articles reporting the impact of chemicals polluting waterways and even causing health problem to the farmers. They are the frontliner or the first one in this regard to be exposed to the high use of chemicals, be it fertilizer or even pesticide. And another issue with chemical fertilizer is the increase in their price. In fact, we have seen an increase up to 30% of the fertilizer price compared to last year. And this is partly due to the ongoing war between Russia and Ukraine. And they are the main producer of agricultural commodities. And like I mentioned just now, farmers can apply more and more chemical fertilizers, but unfortunately, not 100% of the applied fertilizer will be taken up by the plant. In fact, findings by Liu 2010 has shown that the fertilizer end use efficiency, in this case is urea, is only 55%, meaning to say that the remaining 45%, close to half of the applied end fertilizer, is lost. How they are lost is through leaching, through soil erosion, through gaseous emission, and this is more prevalent, especially when you apply the end fertilizer during rainy season or on hilly terrain. So not every single fertilizer that you apply is being taken up by the plant. In this case, it's only 55%. So what happened to the excess N and P fertilizers? Through surface runoff, through leaching, they are being leached out to the nearby water sources, becoming a source of food for the algae, and this results in the algae bloom, increased overgrowth of algae in the water, the biological oxygen demand will increase, and as at the end result, the population of aquatic life will be reduced dramatically. So this phenomena is called as eutrophication, which can be seen happening in our lakes, rivers, and so on. So as the alternative solution for that, we could optimize or make use of the soil-plant microbe interaction, or in other words, we should start promoting the use of biofertilizer. And this is also just recently, just a few days ago, a local newspaper, NSTP, has quoted the Minister of MOSTI saying that they will start to promote the use of biofertilizers and technologies to increase the crop yield. This is partly due to the rising cost of fertilizers due to a global supply shortage of raw materials. So I think this is the best time and we are on the right track for us to start promoting soil microbes for sustainable agriculture. And so those are the introduction and justifications on why we choose to work with soil microbes. So in this session, there will be several topics on keys to the success of using biofertilizer in agriculture. First, of course, we need to understand, we need to know the different types of microbes available in our soil. What will be their benefits to agriculture? What are the best application methods to soil and crop? The quality control and quality assurance of a biofertilizer product. And we will look at some of the biofertilizer products currently available in the market and what are the biofertilizer scenes in Malaysia. So the first one, to fully utilize the soil microbe, first we have to know what are the commonly available microbes in our soil and what are their main characteristics. And when we talk about soil microbes, the two most important microorganisms is bacteria and fungi. The first will be bacteria. Bacteria is the most abundant. In 1 gram of soil, in 1 gram of tropical soil, it can contain up to 10 to the power of 8 or 100 million of colony of bacteria per gram of soil, okay? But in terms of masses, in terms of volume and weight, fungi are the highest. This is because they have the extended network of hyphae and mycelium. So most studies involving biofertilizers relies on these two soil microbes as their source of organism. Just briefly about bacteria, it is most abundant in soil. Bacteria is a single cell or known as prokaryote. So far, more than 400 genera has been identified worldwide with more than 10,000 of species. They came in different sizes, color, and shapes. We have the most commonly is bar shape, like for example, bacillus. We have also in the square shape, like for example, coccus, and so on. So the good, of course, we have a good and bad bacteria, but the bad one will cause disease to our crop, and it is known as pathogen. The good soil bacteria are known as plant growth promoting bacteria or rhizobacteria, depending on the on where the bacteria resides. Okay, these are the groups of soil bacteria that have been proven to benefit the plant through various mechanisms, and they can be grouped into gram-negative and gram-positive bacteria, like for example, Pseudomonas, Bacillus, and Azospirillum. Another group of soil microbe is fungi. It is a multicellular or eukaryote. More than 70,000 species are known. It is high in biomass because they have the extended hyphae and mycelium. They usually can be found in area with high organic matter, especially in tropical forests. They require oxygen to survive, and they are more resistant toward environmental stresses, like for example acidic soil, because of the external structures of hyphae and mycelium. And the good soil fungi can interact with plant roots, and this relationship will benefit the both parties, and it is known as mutual or symbiosis relationship. How they do that? The fungi will help the plant to absorb more water and nutrient, and in turn, the plant root will secrete exudates as the food source for the microbes. Okay, and the fungi that interact with the plant roots beneficially are called as mycorrhiza. There are also other soil microbes, like for example, actinomycetes. Actinomyces is actually a kind of bacteria, but with filamentous bacteria. We have also algae, as you can see at the bottom left picture, algae overgrowing in the lakes somewhere in China. We have also soil fauna, includes earthworm, mites, nematode. Nematode is at the bottom picture. Nematode looks like earthworm, but a lot smaller in terms of their size. We have also protozoa. Protozoa came in different size and shapes. So there are many types of soil microbes, and we have to know and choose those that can benefit our plant. So we will look into what will be the benefits of soil microbes in agriculture. And a product consisting of beneficial soil microbe is known as biofertilizer. The definition of biofertilizer, according to Dr. Zulkifli, is the use of known soil microbes to benefit the plant, to increase the growth, yield, and tolerance towards environmental stresses and pathogens. So biofertilizer contains a known soil microorganisms. There are also another terms when we talk about soil microbes that we call as effective microorganisms, or EM. EM is a various blends of random and commonly found microorganisms. So the main difference between biofertilizer and EM lies in the microorganism itself. For biofertilizer, usually the microbes were carefully chosen and the identity is known. Whereas for EM, it can contain up to 80 types of random soil microbes. Still, both serve the main purpose, which is to benefit the plant, but perhaps with different approach and pathways. So how do biofertilizer consisting of bacteria work? These are their main common mode of action. First, they can fix atmospheric nitrogen, and this kind of group of bacteria is known as nitrogen fixing bacteria. They can solubilize the fixed P and K in soil. These groups are known as phosphorus and potassium solubilizing bacteria. They can also produce phytohormone to enhance the growth of root of the plant by producing acetic acid and so on. They can help to enhance the nutrient absorption, to reduce the chemical fertilizer usage. They can also act as biocontrol of pests by producing antibiotics, by producing siderophore. They can also bioremediate the contaminated soil and also helps in decomposition of organic matter. And when we talk about beneficial microbes and biofertilizers, one of the most common benefits is the ability of the bacteria to fix nitrogen. So very briefly, we are going to look at the nitrogen cycle. We have been blessed by having a tremendous amount of nitrogen in our air. In fact, 78% of our air is actually nitrogen. But this kind of nitrogen is in the form of stable nitrogen gas, held by a strong triple bond. So it can't be broken down easily. It requires a high energy to break this triple bond. The first one, it can be broken down through the natural lightning process. Secondly, it can be broken down through an industrial chemical process known as Haber-Bosch to produce ammonia and urea synthetically. Another method that we are more interested in is through the fixation by the nitrogen-fixing bacteria. So how a nitrogen-fixing bacteria work? This kind of bacteria produces an enzyme called as nitrogenase enzyme. This particular enzyme can break the triple bond of the nitrogen gases into a simpler form, which is ammonium ion that can be taken up by the plant. And this is a common ability of soil bacteria, such as from the genus of Bacillus and Rhizobium. This is the example of rhizobium nodule symbiosis. When we talk about nitrogen fixer, the most effective nitrogen fixer are rhizobium. And these microbes can be found in the nodules of any legumes, like for example, beans, mimosa, peanut, and so on. But they require a specific host plant in order to efficiently fix nitrogen. And the best example of utilizing rhizobium as nitrogen fixer is at our oil palm estate. You will observe a kind of wheat plant growing surrounding the canopy of the palms, but this is actually not an ordinary wheat. It is actually a legume called as Mucuna bracteata. This practice is called leguminous cover cropping, which has various benefits to the soil as well as to the palm. And the most important benefit is the legumes will help to fix nitrogen from the air and supply it to the crop. And beneficial soil microbes can also solubilize the fixed P and K as described in the earlier slide. And how do they do that? PSB has the ability to secrete phosphatase enzyme as well as organic acid that can solubilize and break the P complex into much simpler orthophosphate. Orthophosphate ions is the form of phosphorus that can be taken up by our plant. Soil bacteria can also produce phytohormone. In this case is acetic acid to increase the plant root growth. So this is another important characteristics that should be taken into consideration when selecting the best soil microbes during the biofertilizer development process. Another important function of soil bacteria is the decomposition of organic matter. So I will show an example of using beneficial bacteria for the decomposition process of organic waste into organic fertilizer product later on. Okay, so how does beneficial fungi work? It can help the plant roots to acquire nutrients, especially phosphorus. How do they do that? Through their extended network of hyphae and mycelium. They can also help to solubilize the fixed P in soil. Similar mechanism or pathway as in bacteria by production of organic acid and phosphatase enzyme. They can help to increase the plant tolerance towards water stress, and this is through the extended network of hyphae and mycelium that can help the plant root to reach a longer distance compared to without the mycorrhiza. They can also act as by control agent. Most important pathway is through the production of antibiotics. They can help to improve soil aggregation as well as producing phytohormone. So this is just a diagram to help you to understand better about how the fungi help to acquire phosphorus. And as you can see, the first picture, there's a gap or zone separating the root hairs with the available phosphorus. But when we introduce our mycorrhiza for fungi, beneficial fungi, their hyphae is helping the plant to reach to that available phosphorus and supply it to our plant. So these are the mechanism or how the fungi can help to acquire phosphorus in the soil. And those are some of the pathways of how bacteria and fungi can benefit the crops. And through a series of simple lab tests, we will be able to determine these beneficial properties as well as other properties, like for example, the production of siderophore, the production of enzyme, and so on. And another beneficial aspect of soil microbes is in biocontrol. And there's a specific kind of bacteria that can help to solubilize, they can help to sort of control the insect and worms attacking our plants. And how they do that, they can produce toxin to kill off those insects and pests. There's also a report by Sundram and also other researchers reporting the effectiveness of using soil microbes to control Ganoderma attacking oil palm seedlings. And this particular report has shown that the combination of bacteria and fungi are very effective to control as much as 57% of the disease of Ganoderma in our palm. But to my understanding, this kind of research, it requires more studies and validation before it can be commercialized and applied on a large scale basis. Use of soil microbes in bioremediation. It can be used in bioremediation purposes to reduce the chemical and heavy metal in contaminated soil. And there's a kind of microbe that can dissolve the systemic herbicide, a very potent and dangerous systemic herbicide, 2,4-diamine, and they can also produce siderophore, and these are the mechanisms. And so those are the common benefits of soil microbes in agriculture. Recently, due to the climatic change and the rise of the seawater level, we started to hear more and more cases of flood and seawater intrusion into the agricultural land, especially to the rice growing areas. This is more prevalent in the northern part of Malaysia as the rice area is situated nearby the coastal line. Once flooded with seawater level, once flooded with seawater, the growth and yield will be reduced significantly as rice plants are sensitive towards high salt content or saline soil. And this will cause more problems to our food security status as mentioned by the Ministry of MAFI in this particular article. So we in our lab, we have come up with strategies to use PGPR with a salt-tolerant ability to reduce the salinity effect on rice whilst trying to improve the cropland growth and yield at the same time. This research was done by one of my doctorate students, Dr. Rakiba. And from her study, based on her study, we managed to determine the mechanisms of how some beneficial microbes could survive under high salt conditions. And the answer is through the production of EPS or extracellular polysaccharide and biofilm. EPS possesses cementing properties to strengthen the cell aggregate, whereas the biofilm protects the cell as a mechanical barrier from the unfavorable soil condition. And the selected bacterial strains, the salt-tolerant strains, were also able to promote the growth and yield of rice grown under saline condition, as can be seen from these three graphs of yield parameters. Using one of the strain, UPMRB9, identified as Bacillus, the inoculation successfully promoted higher yield parameters, and this study, in my opinion, could serve as a preliminary basis to utilize salt-tolerant microbes on a large-scale basis in agricultural areas affected by seawater intrusion. And the third topic in this session is the best application methods of biofertilizers to soil and crop. Biofertilizers can be grouped into two forms, solid or liquid. If to develop a solid biofertilizers, we require carrier, like for example, an empty fruit bunch, palm oil meal effluent, and so on. And normally, the suitable carrier is sourced from agricultural byproduct. How to apply a biofertilizer? Biofertilizer application method. It actually depends on the types of biofertilizer and crop of interest. The main objective is to ensure that our applied inoculum could reach the plant roots as soon as possible after application. And why is that so? Because the plant root secretes exudate, which becomes a source of food for the microbes. So the best will be to spray or broadcast the biofertilizer product on the soil surface. We can also apply biofertilizer by coating the seeds or applying them on the planting bed just before the seed sowing. And this is a picture showing some of our previous studies using biofertilizer mixing with the rice seed before sowing. We have also tried applying biofertilizer in oil palm by spraying on the soil surface, mixed with the fertigation system. Trials of biofertilizer in the rice field at Kelantan and Malacca. And the fourth topic is the quality control of a biofertilizer product. And I think this is one of the most important aspects during the development and commercialization so that our products remain effective in the field, not only under controlled lab conditions. This is related to the earlier point of getting farmers' confidence to use soil microbe as an alternative to chemical fertilizers. At the moment, we do not have any biofertilizer act or regulation, so everyone can come out with a biofertilizer product with all the claims. And how to choose suitable and effective microbes for crops? First, we have to select and isolate bacteria and fungi with broad host capabilities. It has to be a non-rhizobium because, as I mentioned earlier, the rhizobium is a specific. It requires a specific kind of host. So we have to choose a free-living bacteria. Those kind of bacteria has to be able to withstand unfavorable stress soil condition, especially in high temperature, salinity, and low pH. How to do that? And some kind of bacteria can form endospore and can be in the dormant state. They also must be able to compete with indigenous microbes. They must able to compete with indigenous microbe and become persistent in the soil because no point if we will be able to isolate superior strains of bacteria, but when we apply to the soil, they are not able to compete with the indigenous microbe. They must be able to be easily cultured on media and a fast grower. The bacteria of choice must be genetically stable and not easy to be mutated. So these are how to choose the suitable and effective microbes for crops. And as a storage and precaution, choose suitable biofertilizer types for the crop. Like for example, if you want to apply in the rice field during the planting, you can choose a liquid biofertilizer because it is easier to do so. If you want to apply during the land preparation step, you can choose a solid biofertilizer. You can just broadcast your solid biofertilizer during the land preparation. The biofertilizer product must be freshly prepared with a shelf life of at least 12 months because, as I mentioned earlier, if you have successfully isolated a superior strain of bacteria, but without proper optimization of the media, the biofertilizer product could not last long, so it has to be freshly prepared, at least with a shelf life of minimum of 1 year. You have to store it in a cool and dry area. Application-wise, we recommend that you apply your biofertilizer product at the different times with chemical fertilizers. And another important aspect related to the use of biofertilizer is the consistency and persistence of the applied microbes around the plant roots. And this is crucial because if they can't survive, the benefits to the plant won't last long. One of the methods is to observe the microbial cell by using a high definition microscope such as SEM and TEM. Another method is by using molecular tagging method, green by tagging the bacterial cell using GFP or green fluorescent protein. And this is rather a complex technique, but as the result, we can observe our applied bacteria at any point of time. And since biological nitrogen fixation by the NFB is one of the main abilities of beneficial soil bacteria, that we need to determine and calculate their efficiencies so that we can recommend the application rate, as well as the possible savings of chemical fertilizers. So how to calculate a BNF rate? There are three methods commonly used. The first one will be the easiest one, comparative calculation. Easily you can do so by comparing the yield and nitrogen content of your control plant versus inoculated plant. The second option, the second method is by using acetylene reduction assay, ARA. This can be done by measuring the nitrogenous enzyme activity, which is by measuring the ratio or rate of conversion of acetylene to ethylene. So this must be done under control condition in the lab. The third method is the most accurate but also most expensive, which is by using N15 isotope dilution technique. This is by using the concept of measuring the differences between applied N15 and N14. So that will be the measurement of the ratio, and this has to be done in collaboration with the Malaysian Nuclear Agency. So we have done that in a study back a few years ago. This is involving the N15 isotope dilution technique on rice plant. We did with collaboration with Malaysian Nuclear Agency, and from the results of the red arrow, you can see the percentage of nitrogen derived from atmosphere is 22%. Meaning to say the mixed inoculum of bacteria of choice has successfully fixed 22% of nitrogen and the required nitrogen of the plant from the atmosphere. So in theory, we could save up to 1/3 of the end fertilizer requirement by introducing this bacterial strain. And we have published that in one of the papers. And another previous study done by Dr. Zakri, one of my senior, also showed the high efficiency of nitrogen fixation in the field trial of young oil palm at one of the Sam Dhabi estate in Johor. So to determine a good quality biofertilizer product, we have to first start with the correct selection of soil microbe, followed by an in-depth study of their benefits, and then we have to do repeated trials and persistency in field condition. Only then we are more assured of their quality and to proceed with the commercialization step. So the last topic of this session will be some insights into the Malaysian biofertilizer scene, as well as some past and present research on biofertilizers conducted at our lab in UPM. This is also one of the study by Dr. Zakri involving the preparation and production of a solid biofertilizer. First, we have to choose the best media for our biofertilizer. In this particular case, he's using a combination of ground empty fruit bunch and also oil palm frond. And after mixing well the media, they pack the media in a polyethylene bag and then send it to the Malaysian Nuclear Agency to be sterilized using gamma irradiation. Then the inoculum production, we have to select the best inoculum or bacterial strains, and then we apply it to the sterilized media. Usually, the minimum bacterial population should be 10 to the power of 6 or 1 million of bacterial cells per gram or per mil of the biofertilizer product in order to be effectively benefiting the crop. So to produce a liquid biofertilizer, similar concept as in the production of solid biofertilizer, we select the best strains. It can be single or combined inoculum. We have to add some additives to the liquid media to prolong the shelf life of the product, and it will be produced on a large scale using bioreactor. So these are some of the examples of our past experiments conducted on various crops to show the effectiveness of soil microbes as a biofertilizer. You can see this is a study involving pineapple on rice plant. We did that on the rice seedling. You can see the difference between the control and the inoculated plant as early as 7 days. And when we prolong the trials up to grain or harvest stage, we can see an increment of yield up to 60%. And this study is with minimum amount of chemical fertilizer, which is 33%. The effect of biofertilizer on root growth of rice, you can see on the right picture, the combined inoculum of two bacterial strains produce better root growth, possibly due to the production of phytohormone. And one of the study, we also discovered that by inoculating the rice plant with one of our bacterial strain, PGPR strain, the rice plant seems to be able to tolerate water stress better compared to the control. And when we determine the soil bacterial population before and after inoculation, we know that after 7 days, the bacteria are growing in our pot. And then the test of the effect on banana plant, the tissue culture. Another important aspect is by control abilities of our bacterial strains, and this is the test done on Fusarium oxysporum, which is the fungi responsible of infecting banana, causing the disease of Fusarium wilt. So without the PGPR strains, the Fusarium fungi, the hyphae, the colony are growing normal on the plate. And when we observe under microscope, we can see the hyphae are growing on a normal hyphae wall, no swelling and so on. But when we introduce our bacteria, when we introduce our PGPR, we can see straight away that the growth on the plate is disturbed. And when we observe under microscope, we can see their hyphae becoming abnormal, swollen and thickened. So we know that this particular PGPR has the biocontrol ability to disrupt, or at least to disrupt some of the stages in the Fusarium wilt on banana. Similar case when one of the post-doc of Dr. Zulkifli, he did the glasshouse trial on banana. He observed similar kind of scenario in which inoculation of PGPR reduced the disease severity index up to 40%. Effect of the PGPR on banana yield with minimum amount of chemical fertilizers, it is better compared to the control. And then there are also other studies involving the use of mycorrhiza or fungi on old palm seedlings. We can see by inoculating the palms with glomus and scutoles spera, the growth are better compared to the control. And when we did the test on the ganoderma infection, we know that we observed that the inoculated palms are better. The test on the effect of sweet potato by one of the previous student. So these are some of the biofertilizer products in the market. And starting from the left picture, it is a rather interesting packaging by All Cosmos or Real Strong, and they name it as a biochemical fertilizer. It consists of chemicals, microorganisms, and organic matter. So this is specifically for durian. We have also bio-enriched M. We have the other product at the bottom, at the middle bottom picture is AG biofertilizer. This is being produced and distributed by a local company called as MLBA, and their target crop is all palm. We have also other products such as BioVita and so on. And there's also one company called as NM BioAgro. They are producing and distributing biofertilizers. And we have been doing research collaboration with Mr. Nazri, the CEO of this company. And for example, they have B-Speed, the biofertilizer product named as B-Speed, which contains phytohormone, nitrogen fixer, as well as nutrient solubilizers. We have also Fusarium. Fusarium is to control the Fusarium wilt on banana. They have also Orchida, obviously for orchids and ornamental flowers. They have also commercializing a product named as Soil Lacto B, which contains lactobacillus. There's also another collaboration ongoing between us at UPM and TP Green Farming Resources with their director, Mr. Chai, and they are the sole distributor of this product from Germany called as Biosols. And this is a kind of biotechnology product that they call it as microbial activator. And through biotechnology advancement, this kind of product it can co-stimulate the indigenous microbe in the soil through a specific signaling process. We have also another ongoing collaboration involving a biofertilizer product called as Bio25. It is a soil enhancer. It contains 25 strains of beneficial microbes. It has been reported to reduce the fertilizer usage up to 30%, increase the yield by up to 60%, and it has been patented by a local company. So the name of the company is Anggun Rezeki Farm Sdn Bhd, and the collaboration between the company and UPM was just started, initiated by the company's managing director, Mr. Raj. There's also biofertilizer products from UPM. On the left is Beto10. It is a kind of liquid biofertilizer developed by Dr. Zulkifli Haji Shamsuddin. And the right picture is B Green. It is a kind of solid biofertilizer being developed by Dr. Razia, and both are from the Department of Land Management, Faculty of Agriculture. And another recent collaboration with us in UPM is by a company called Syarikat Hajimat. And through the collaboration, we have successfully come up with a patented liquid biofertilizer called as Smart Enzyme UPM. And by using that particular enzyme and microbes, in combination with animal manure, we produce an organic fertilizer product called as Milagro Premium, which are now available in Malaysian market. Some information about this particular product, Milagro Premium. It consists of up to 2 million of CFU colony forming unit of bacteria per gram of the product that have been proven to increase the growth and yield of various crops. It contains additive amendments to optimize water and nutrient absorption by the plant roots. So these are the technology has been patented. And one of the benefits of the product is the shelf life can goes up to nearly 2 years, stored under room condition. And another important advantage of this product is they are able to form the particular microbes in the product are able to form endospore whenever they are being exposed to harsh and extreme condition, like when the food are being depleted, the oxygen is being depleted, and so on. The bacteria has the abilities to form endospore so that they can survive longer under dormant state. Some nutrient analysis of the particular product, Milagro Premium, we know that this particular product, even though it is being made by animal manure, but it does not contain any pathogenic microbes. They contain a fair amount of NPK as well as additives. Trials being done by the company on long bean, we can see by looking at the pictures, by the Milagro Premium product produce 50% of better overall plant growth compared to the chemical fertilizers. There are trials on cabbage. We can see by using that particular organic fertilizer, it produces better yield. And there's a lot of customer testimonies that you can find in their official website. And if you need more information, the Malaysian PGPR Society, in collaboration with Faculty of Agriculture UPM, is going to organize the 7th Asian PGPR International Conference for Sustainable Agriculture next month. So you can have a look at our conference site because our registration is still open, and there are a few companies that I mentioned earlier, like for example, All Cosmos and Syarikat Hajimat producing the Milagro that will be joining us in this particular conference. And if you need more information about Milagro, if you want to meet the Milagro team, you can do so at Maha 2022, scheduled to be on 4th of August at MIP Serdang, Selangor. So as the conclusion, bio and organic fertilizers are relatively cost-effective green technologies for increased crop yield and sustainable agriculture. But in reality, we cannot fully replace the chemical fertilizers with this kind of bio and organic fertilizers because we know that bio and organic fertilizers does not contain nutrient as high as much as chemical fertilizers. We still need chemical fertilizers in order for a sustained and longer period of cycles of agriculture, but perhaps with a minimum rate or minimum amount. So the keys to success to utilize biofertilizer or soil microbes, first, just for as a recap, we need to understand the needs of the soil and crops. We need to understand, we have to have the knowledge of what are the benefits of different types of soil microbes. As I mentioned earlier, what can be done by the soil bacteria, what are the benefits of soil fungi, and so on. Secondly, we need to find the optimum application method so that it will be more effectiveness and effectiveness, meaning to say the bacteria or the soil microbes has to be able to infect the plant roots and it has to be effectiveness. It has to be producing the beneficial effects to the plant. It has to be persistent. It has to be able to compete with the indigenous microbe and stays under harsh condition. So the third point is the quality control of a biofertilizer product. This is very important. First is the most important one is the shelf life. The biofertilizer product must have a longer shelf life in order to be commercialized. And the fourth one, the keys to success is the biofertilizer scenes in Malaysia. How we can integrate the biofertilizer application, the soil microbe technology with the conventional farmer practices. As I mentioned, we can do so, but maybe we have to have a little bit of gap of time between the application of biofertilizer and chemical, be it fertilizer and pesticide, and what will be the potential for future improvement. So I think with that, I would like to end the session by this sentence: beneficial soil microbe, the future approach to food security. So I would like to thank my mentor, Dr. Zulkifli, Dr. Razia, and also our collaborators, Milagro, Malaysian Nuclear Agency, Biosols, Anggun Rezeki Farm, and NM Bio. With that, I thank you all for your attention. Pass it back to Puan Atira.
Okey, terima kasih. Terima kasih Dr. Ali. Jadi sekarang kita buka untuk ruangan soalan. Okey, soalan pertama. How effective are commercial biofertilizer that is dosed with added microbes? Are their claims exaggerated?
Okay, thanks for the question. Yeah, as I mentioned earlier, we have there is no act or regulation to control biofertilizer products in Malaysia. So every company can claim whatever that they have, but yes, if someone with knowledge, we can take the product and then we can determine or analyze in the lab so that we know what will be the exact microbes in that product and what are their claims. It's no doubt, I'm not denying that their claims are sometimes exaggerated or more than what the microbes are capable to do so. Yes. Puan Azira?
Soalan dari Siti Hajar. In Malaysia, which country use this biofertilizer? Maybe
I think I could not understand the question. Which country use which fertilizer? Maybe Siti Hajar can describe that question in more details later on. Do we have any other question? Okay, yes, regarding this particular question, of course, we want to encourage home gardeners to start producing their own so-called microbial product, and the best will be by a simple fermentation technique to produce effective microorganisms using the bioproducts from their kitchen, the bioproducts from the farms, and so on. They can do so, but as I mentioned, the quality control is one of the main concern because, yes, as mentioned by Dr. Chris, there is a risk of accidentally introducing bad microbes because in the effective microorganism product, we are supplying the molasses, the sugar content as a food source for all the indigenous soil microbes. So we know that in the soil there are also bad microbes as well. So and the issue is regarding the quality control of that home produce. Okay.
From Rakibah Sultana, how much amount of biofertilizer is required to apply in rice field?
Okay. This is actually depending on the variety of the rice as well as the condition of the field. First, we have to determine the the soil fertility status, and then it depends on the kind of biofertilizer as well. The biofertilizer must be able to at least supply some or if not all of the required nutrient by the crops. So I think if I'm not mistaken, somewhere between 3 to 4 tons of solid biofertilizers are required per hectare of rice planting areas. But that is a very generic recommendation. So it requires a more specific trials and details before we can recommend further. Okay. Yes, I totally agree with Prof. Shen because in my opinion, the organic fertilizer alone will not be able to meet all the nutrient demand by the crop. Okay, because we know organic fertilizer it contains a very relatively low amount of NPK at least. And for biofertilizer alone, biofertilizer from the definition itself, it does not contain any elements of NPK, but rather it contains the microbes that helps to solubilize the NPK already available in the soil, helps to fix nitrogen from the atmosphere and so on. So biofertilizer is actually it does not contain any NPK, so we still need chemical fertilizers, maybe perhaps in a minimal amount. So in our case, in our previous study, normally we apply biofertilizer or any product of some microbes with at least 30% in combination with 30% of chemical fertilizers.
Question from Pisang, Alto Panjang. For homemade fertilizers, such as compost, what test would you recommend? Does UPM offer the service?
Okay, if you want to test the quality, if you want to be sure of the quality of your homemade fertilizers or biofertilizers, the test, if it is in the form of compost, of course, the test will be the nutrient element. We want to know how fertile or what are the quality of the fertilizers. We have to know how much the nutrient element inside that particular product. But if you want to talk about biofertilizers, the first test will be we want to determine what will be the total microbial population contained inside that particular product. As I mentioned in the slide earlier, if you are talking about soil, if you're talking about bacteria biofertilizer, we have to have a minimum population amount of 1 million or 10 to the power of 6 CFU per gram of the product in order for them to be effectively benefiting the crop when applied later. Yes, our department in Faculty of Agriculture do offer that kind of services. First is to determine the nutrient element. Second is to determine any microbial parameters. Or if you want to determine very specifically the identity of the microbial, microbes, then we can also offer that kind of services as well.
Another question from Pisang. Is there any short online courses related to regenerative agriculture in Malaysia?
As far as I know, we did not offer that kind of courses. We do offer a basic soil science courses, but that one is physical courses, is a face-to-face courses.
From Nazifa Saimi, is there any challenges in the implementation of biofertilizer, like the consistency of these microbes under field condition?
Yes. I've been mentioning in my slides, it is very difficult. One of the challenge that we are facing is to gain confidence by the farmers to start using biofertilizers because they cannot see what they are applying to their farm. Okay, and another reason is because the effect are usually much slower compared to applying chemical fertilizers. So those are the two main challenges in the implementation of biofertilizer. Apart from that, another challenge is the quality of the so-called biofertilizer. If you just buy any biofertilizer in the market and you apply it to your plant, and then after some time you observe there are no benefits of applying that biofertilizer, it is partly due to the quality control of that biofertilizer itself. So yeah, when we talk about biofertilizer, because we are dealing with living things, there are a lot of challenges. Another one is related to the shelf life of the biofertilizer. Because of course, living things have their own shelf life. Let's say if you buy a biofertilizer product, they claim so and so they can do a lot of stuff, but actually because of the shorter shelf life, you are actually buying biofertilizer containing dead microbes. So there will be no beneficial effects when you apply to your plant later. Okay.
Okay, another question from Pisang. I came across some interesting publication by Dr. Ali on biochar. Will you comment on any way to determine the quality of commercial biochar products? What is the difference between biochar and charcoal?
Biochar and charcoal, the difference is the storage, the temperature. If the temperature is too high, it will become charcoal. If the temperature is, if I'm not mistaken, between 400 to 500, it will become biochar. And the preparation is also different. Biochar is under the condition without oxygen or minimum amount of oxygen. Charcoal is just a direct burning process of the log and so on. Okay, so that will be. I'm not an expert to comment much on biochar actually.
From Abba Nabai, some of these beneficial microbes do also have pathogenic attributes. Why we often call them beneficial microbes? Is it because their benefits outweigh the pathogenic effects?
Okay. To my understanding, if the microbes contains characteristic that can be regarded as pathogenic attributes, it is safe to say that it is safe for us to find or select other strains because we don't want microbes that can be benefit and also at the same time can cause negative effect to our crops as well. So you can do your characterization, try to find those microbes that only benefit the crops, but without all those pathogenic attributes. Okay, I think that hopefully can answer that question. Because regarding the regulation by Department of Agriculture, there are also they have also categorized some bacterial strains. In this case, is bacteria that are pathogenic. So if your product contains this kind of bacterial strains, like for example Bacillus cereus, of course Bacillus cereus can be benefit to your crop as well, but DOA label it as pathogenic bacteria. So if your product contains Bacillus cereus, even though your product is very good to the plant, but you cannot sell it and market it.
From Odin Sinin, what is the shelf life for this biofertilizer?
Which one? The biofertilizer that I mentioned, there are a lot of examples, but when we talk about the Milagro Premium, based on our studies, the shelf life of the combined beneficial microbes inside, it can goes up to 23 months or nearly 2 years. So that will be the shelf life. But to be safe, because the study is being done in under controlled lab condition, to be safe when it is being produced commercial in the market, we put it as 1 year, the shelf life of the product. Okay.
Okay. From Fauzihan Karim, I am sure if all claim microbes are functionable in all conditions, what is your view on this? For example, I do not think if all claim microbes can survive in acid sulfate soil, has had the microbe commercialized were not originally from there. Or do you think nowadays we have specific microbe for acid sulfate soil?
Uh, yeah, I think I can answer part of the question because part of the ongoing research by my master student is she's trying to find the best several strains of PGBR to be effective on acid sulfate soil. So based on her study, ongoing studies, she has found out that this particular some of the strains of bacteria are able to survive under acid sulfate soil. There are a lot of mechanisms involved, and they can also benefit the crop at the same time. But not every microbes can be beneficial under this particular very specific soil condition, as you mentioned. The companies producing biofertilizer can claim everything, but yeah, we as a researcher we have to determine in detail through analysis in the lab. So, as I mentioned, the student, based on her findings, she managed to get 2 or 3 strains that can survive under acid sulfate condition through various mechanisms, but the other strains are not doing well. Okay.
From Sulaiman Hamzat, kindly discuss on the specification of users in grasses.
It will be almost the same like in rice, okay, in grasses, rice, and so on. But we have not so far did any research very specifically on grasses. But theoretically, you can use the same rate to start, the general recommendation rate to start with. It will be almost the same like rice or paddy.
Saya rasa itu soalan yang terakhir rasanya. Jadi kita boleh rate out. Okey, Assalamualaikum warahmatullahi wabarakatuh dan sekian sahaja perkongsian kita pada hari ini. Semoga berjumpa lagi di webinar akan datang insya-Allah. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera.
Thanks everyone.